A very warm welcome to the participants of the virtual international conference on basic and translational cancer research on novel ideas and approaches. And today my area of my presentation will hopefully fit on the novel therapeutic themes of this conference agenda. Before start my presentation, I would like to express my thanks to the organizing secretary of this conference, Professor M Lata, and the coordinator of Dr. P. Ashok Kumar, Associate Professor, and other faculty members for organizing this conference and giving me an opportunity to share our research ideas in this forum. So the title of my presentation is Targeting the FOXO3 Signaling by Bioactive Natural Product in Breast Cancer Prevention. We all know the FOXO3 is an important factor and is also act as a tumor suppressor, and it is play important role in mediating the variety of cellular process including apoptosis, proliferation, cell cycle progression, DNA damage and tumorigenesis. So in this presentation, what I am going to present is the importance of the FOXO3 signaling in cancer chemoprevention and their associated downstream signaling pathways such as PI3, AKT and other programmed cell death active pathways. And important drug aspect which I am going to talk about is a bioactive natural product which has been isolated from the cumin seeds. In botanical name we can call it as a Centantum athalmeticum seeds. It is called Kala Ziri in Hindi. So one of the active fraction from this seed is called as a vernodolin. That is the active component which we are going to talk about as a drug in this presentation, and how they able to prevent the breast cancer prevention through in vitro and in vivo detailed activities by using various research experiments. Now let us move on to the let us discuss more about the understanding the breast cancer. I hope in this conference forum most of the research scholars and graduate students are able to participate. I would like to give a brief outline about that, how that breast cancer has been happening in detail. In detail, cancer occurs as a result of a mutation or any abnormal changes in the genes responsible for regulating the growth of cells and keeping them healthy. Whereas if you take as a cancer mutation can turn on certain genes and turn off others in a cell, that changes cell gains the ability to keep dividing without a controlled manner or producing more cells just like it and forming a tumor. So this is the structure or figure will show how the normal cancer cell process has been happening. If you see the normal cell get mutated, the cells cannot repair itself and it damages and cells multiply without control. This is the simple cancer cell process that has been happening. A tumor can be defined as two aspects. One is a benign tumor that is not dangerous to the health. On the other hand, it is called a malignant, has the potential to be dangerous. These are a very basic concept where this benign tumor grows slowly and they do not invade nearby tissues or spread other parts of the body. Whereas if you take the malignant tumors or cancerous, if left unchecked or untreated, malignant cells eventually can spread beyond the original region of the tumor that has been generated, and it also spreads over the other parts of the body. If you take the breast cancer, it is defined as the most prevalent disease and hold second rank in the mortality rate of women after the lung cancer. So the recent data suggests that in 2017 approximately 281,840 new breast cancer cases and more than 50,000 breast cancer deaths are estimated throughout the worldwide. Cancer, it's defined as in situ, that is meaning as in place, or other hand invasive or infiltrating to the other region. So if you take in specific to the breast cancer, this is the normal duct where you can able to see that normal ductal epithelium has been surrounded along with the myoepithelium on the outer region. If you take the ductal hyperplasia condition of the breast cancer, it is also called as a ductal epithelial hyperplasia. It happening as an overgrowth of cells that line the small tubes, that is called ducts, inside the breast. If you take the atypical hyperplasia, is a precancerous condition that affects cells in the breast. This atypical hyperplasia is not a cancer, but it can be forerunner to the development of the breast cancer in future. The other case is said to be DCIS. It is called as a ductal carcinoma in situ. So the meaning itself, the cells that line the milk ducts of the breast have become cancer, but they have not spread into the surrounding breast tissues. So DCIS is considered as a non-invasive or pre-invasive breast cancer. The final one, it is most prevalent cases of the breast cancer. It is called invasive ductal carcinoma, IDC. It is simplified called, and it is about responsible for the 80% of the all breast cancers or invasive ductal carcinoma stage. This IDC starts in the cells that line a milk duct in the breast. From there, the cancer breaks through the wall of the duct and grow into the nearby tissues. So these are the different stages of the breast cancer condition that has been happening. So this slide will show the percentage of new cases by age group in all races. These all races which include Asian, then American and Western countries region that has been happening in the female breast cancer. And if you see the bar graph, from the age group of above 45 are the more predominant, starting from 20.1% of new cases. In the age group of 55 to 64, it shows 25.6%. In the age group of 65 to 74, showing about nearly 25%. It was statistics of breast cancer study on 2019. If you take the ICMR report of breast cancer in India that has been published on 2015, more than 25 years of back, now the present scenario, that's the red graph, which shows the more number of breast cancer patients has been increasing. It has been starting from the age group of 30 to 40, from 7 to 16, whereas in the 40 to 50 case of ages, it has been increased to 22 to 28%, whereas only the 50 to 60% the percentage has been slightly decreased from 36 to 30. So it has been clearly indicate the breast cancer in humans in India, it has been starting from the early ages between 40 to 50 has been evident. Next we move on. What should be the risk factors for breast cancer? We are all know gender, female is the major cause for the breast cancer, and a very rare occasion 1% males has been happening. And the race, as I told, based upon the color, more common in whites as happening in other European and other region of worldwide. And if he increases the age, increases as a woman gets older. This is the common factor. And a relative such as mother or sister or any family history that is having a breast cancer have the possibility. And also the menstrual history that has played important role during the early onset of late menopause. And childbirth, the first child after the age of 30 or having no children at all have been the cases of the risk factors. Apart from that, what are the physiological activities that has been associated with the risk factors for breast cancer, which include obesity, diet that has fat and alcohol consumption, too much of consumption, and the lack of physical activity and stress, radiation exposure, especially the persons who are working on more on X-ray, and the history of cancer, which is include the family, breast, uterus, cervix and ovary, and the hormones which include estrogens in hormone replacement therapy and birth control pills, and genetics, certain conditions that are inherited from the birth, and more than 70% of the patients have no risk factors at all. Now coming to the diagnosis aspects which you take, the stages of breast cancers, when we go to the doctor physicians, they classify breast cancer according to the stages, that is the symptoms which we have. So they are done different tests, determine the size of the tumor and areas it has been spread. Based on this, the stage 0 is said to be a least advanced stage, where the cancer cells are within the duct and have not invaded into the surrounding fatty breast tissues. This can be treatable. Whereas it has been next stage is said to be stage 1, 2 and 3, The cancer is distinguished by the size of the tumor and whether it has spread to the chest wall or skin or to lymph nodes near the breast. Whereas the stage 4 is said to be the most advanced stage, the cancer has spread as a metastasis to other organs. It is called metastasized condition to the other lymph nodes far from the breast. So based on this diagnosis, what type of treatment they can do based upon the receptors that has been present on the breast cancers. So first category is said to be a receptor positive, that is hormone receptor positive, where the breast cancer cells that have a high proportion of estrogen or progesterone receptors, the hormone signals cells to increase the cell growth. Whereas breast cancer cell that have a lower number of estrogen or progesterone receptors said to be a hormone receptor negative. On the other hand, HER2 positive breast cancer is defined as these breast cancer patients that they test positive for a protein called human epidermal growth factor receptor 2, that is called HER2, which promotes the growth of the cancer cells. It tends to be more aggressive than other type of breast cancers. The final one is the most worst condition is called a triple negative breast cancer, where the breast cancer patients that test negative for estrogen receptor, progesterone receptor and human epidermal growth factor receptor, it is characterized as more aggressive and less responsive for any standard treatment. So these are the different diagnosis aspects, and based upon the hormone receptors they will do the different treatment aspects. Now coming to the actual, what is the local therapy that has been used for the treatment of breast cancer is called lumpectomy along with the radiation process. The other condition is called mastectomy, whether we can use the radiation or without radiation depending upon the advanced stages of the patients. So the ultimate goal for the local treatment is said to be a primary site of disease. The second stage is called a systemic therapy, where the important role, the painful therapy which has been used as a chemotherapy, hormonal therapy and targeted therapy. Especially if you take any surgery aspects, either lumpectomy or mastectomy has been carried out. Radiation therapy, as I told, by using chemotherapy along with some drugs targeting particular area of the body, and hormone therapy by drug using a commercially available drugs for breast cancer, tamoxifen and aromatase inhibitors, and also the biological therapy that use drugs Herceptin along with the monoclonal antibody receptor positives such as HER2 and lapatinib and metastatic HER2, avastatin and low dose of aspirins. So even though these different type of treatments with the various commercial available of drugs has been available for breast cancer, the ultimate aim and until the end is ending with different side effects that affects the other parts of the body also. So that's why there are several investigation has been carried out on the plant derived natural compounds. This slide we clearly shows the natural occurring plant based anti-cancer database that has been taken out from NPSAT website on 2016. There are several cancer activities has been carried out on these plant derived compounds. Out of this, breast also there has been nearly 500 plant derived compounds has been effectively showed as the chemotherapeutic effects on the breast cancer cells, either from plant derived source or any naturally occurring compounds. Apart from the breast cancer, there are several other plant derived components has been used for several cancers as listed here. So this is some basic idea which will give some food plants having the anti-cancer properties, where these are the day-to-day activities we are taking regularly in our life, either in the form of fruits or vegetables, where the carrots are said to be rich in carotenoids and the grapes are rich in the resveratrol. These are the active components that has been enriched in these fruits or vegetables, and lycopene that has been present in the tomato, and gingerol and shawol is an active component that is present in the ginger, and allicin is the major component present in the garlic, and spinach, the green leaves, has been natural antioxidant mixture, and the indole carbinol has been present in the cabbage, and vitamin C rich in the citrus fruits, and amodine has been rich in aloe vera, and mircitine has been present in the cranberry fruits, and ellagic acid has been more dominant in pomegranate, and eugenol has been present in the cloves, and other components such as sulforaphane, carnosol and vanillin has been present in the other plant derived components. Have been already shown several anti-cancer properties that has been documented by the various researchers. So if you take more about breast cancer on this topic, there has been US approved FDA drug components that has been approved, such as two type of drugs, taxanes, that is called paclitaxel and docetaxel, that has been approved in the year of 1972 by FDA. These paclitaxels are coming under the class of vinca alkaloids such as vinca rosea, vinblastine, vincristine and vindicine and vinorelbine. This is the picture of this plant. This is the mode of action, how these vinca alkaloids has been acting on the cancerous cells. So if you take this A, it is called a normal mitosis picture where the metaphase has been happening with the chromosome and spindle frame formation, where this important molecule is called tubulin molecule that has been stacked to form the mitotic spindle along with this anaphase space is happening. Where in the case of cancerous condition, these mitosis blocked by the vinca alkaloids, especially on the tubulin formation. Without the tubulin formation fail to polymerize in the presence of vinca alkaloids, that results in the dissolution of the mitotic base, leads to the cell death. So this is the process which has been already, mechanism has been derived in the year of 1972 itself for the breast cancer that has been used. What is the mode of the maximum anti-cancer target agents if you take is called as a cell death process, that is otherwise called as apoptosis or programmed cell death. In upcoming slides we will see more about how the programmed cell death has been happening. What is the relation between programmed cell death and ROS? Apoptosis or programmed cell death is a regulated process that allows a cell to self-degrade in order to eliminate unwanted or dysfunctional cells from the body. The biochemical features of cell death process are high molecular weight DNA fragmentation into oligonucleosomal ladder, phosphatidyl serine externalization, and proteolytic cleavage of number of intracellular substrates. One of the important process of the cell death process is the mitochondria, where the generation of ROS is happening inside the cells, which majorly serving as an inducing signal for apoptosis. So this picture will give a clear idea what is the two type of cell death. This is the basic idea which are happening, necrosis and apoptosis. Whereas in the necrosis only chromatin clumping is happening, cellular organelles and flocculent mitochondria has been happening, which results in the disintegration and releasing of the intracellular contents that will form the inflammation. Whereas in the case of apoptosis or programmed cell death, the chromatin condensation and segregation, condensation of cytoplasm, which results in the nuclear blibbing, that is called nuclear fragmentation happening with apoptotic bodies, where the process called phagocytosis, where the apoptotic bodies has been extruded out of the cell of the phagocytic cell. So if you talk about the apoptosis and ROS, there are two important pathways that we have to discuss. One is the intrinsic pathway or extrinsic pathway. In this intrinsic pathway is mainly carried out by the BH domain, which is called the BCL2 domains factor, that is notably involved in the regulation of the apoptosis. What happened in the intrinsic pathway? The growth hormone withdrawal, DNA damage, any UV or gamma radiation, or any anti-cancer drugs, for example we are using for chemotherapeutic drugs, that will result in the inhibition of propeptotic factors such as BEAM along with BCL2 and other back and binding family protein, that will result in the activation of cytochrome C release from the mitochondria into the cytosol. These cytochrome C will activate the caspases. The caspases are nothing but the cysteine aspartic proteases. This is the family of proteases, that is enzyme proteases or enzymes that plays important role in the programmed cell death. This is the one type of pathway where this procaspases activate caspase 9, and these other executioner of caspase said to be 8 and 3 will lead to the cell death process or apoptosis. So these yellow dots will indicate the promote the release of cytochrome C or propeptotic factors, and the pink one which inhibits the release of cytochrome C and anti-apoptosis. This is called as the intrinsic pathway. The other pathway is called as the extrinsic pathway, where the DR is nothing but that's the death receptors has been binding within a specific adapter protein that is called FAD. FAD is nothing but Fas associated protein with death domain, as because this death domain bind with the death receptor, that will result in the procaspase activation into caspase 8, and the caspase 3 will be activated. As I told here, the intrinsic pathway, the similar mechanism has been happening at the end of the process. Now coming to the what is the link between the FOXO3 and PI3-AKT downstream signaling in the cancer condition, we will discuss detail in this slide. As I told in the introduction, forked box is a transcription factor, belongs to a superfamily called winged helix DNA binding domain. These FOXO functions mainly cell proliferation, cell cycle progression, cell differentiation, tissue homeostasis, angiogenesis and apoptosis. So what happens? The deregulation of these FOXO transcription factors can lead to the severe pathologic condition that includes the cancer. There are different types of FOXO factors, that is FOXO1, FOXO3, FOXO4 and FOXO6. These FOXO1 and FOXO3 expressed in all tissues nearly present in our body. In our study we are going more focused on the FOXO3A. And FOXO4 is majorly present in the muscle, kidney and colorectal tissue, and FOXO6 especially present in the brain and liver. Now coming to the our FOXO3 functions a downstream pathway of ERK, MAP kinase, PI3-AKT kinase and IKK pathways, and therefore facilitate the crosstalk between the three frequently deregulated signaling cascades in most cancers. Anti-cancer drugs such as commercially available moxitaxol, doxorubicin, lapatinib, gefitinib, imatinib, cisplatin and tamoxifen are mediated through FOXO activation primarily via inactivation of the PI3-AKT axis signaling. Now coming to the basic aspects, how the nuclear transport and cancer from mechanism to intervention has been happening. This picture will clearly show two pictures. Picture A, where the normal cell, which has been anchored by the insulin-like growth factor, epidermal growth factor and common gene called ER2, ERBB2, that is called receptor tyrosine kinase. So in normal cells, what happens? The PI3K activation can be inhibited by the P10, that is called phosphatase tensins. This results in the nuclearization of FOXO transcription factors into the nucleus. In the nucleus, these FOXO3 can activate transcription of genes, promote cell cycle arrest and apoptosis. These are the regular process that has been happening routinely. So what happens is P27 complexes with the cell cycling related protein CDK2 and cyclin to inhibit the E2F2 mediated transcription that leading to the cell cycle arrest. So this is the controlled process that has been happening in the normal cell, as I told in the picture A. Now coming to the cancerous condition. In carcinoma cells, the picture B, whether the lack of P10 activity, the PI3K-AKT signaling is active, that results in the phosphorylation of the FOXO protein and P27 by AKT. Now the FOXO phosphorylation promotes its nuclear exports, that is called CRM1, that is called chromosome regional maintenance 1 dependent manner, so that it cannot be no longer or actively involved in the transcription of the target gene. So now the AKT also phosphorylates the P27, disturbing its nuclear localization signal and forcing to remain in the cytoplasm. Now the CDK2 other cell cycle related protein is no longer inhibited and is free to activate the E2F1 transcription factors that will lead to the ultimate cell proliferation. So this is how it may be varies between the normal cell and cancer cells. So this slide, which has been published recently in 2015-16 from Clinical Cancer Research, show the clinical anti-cancer treatment target FOXO3 through 3 oncogenic kinases, as I told in previous slides, AKT, IKK and ERK, not only from the breast cancer, other related cancers such as myeloid leukemia, osteosarcoma, prostate, kidney, ovarian, leukemia. If you see this from the cancer type, these are the commercial drugs that has been used. Most of this protein that has been targeted for the chemotherapeutic or chemo prevention aspect is said to be a FOXO3 protein, and most of the downstream pathway that has been reported is AKT, ERK, and some of them are still working on. So these are the signal pathway that has been affected, which has been shown detailedly on these different type of drugs that has been used during the different type of cancer cells. Now coming to that our FOXO3 pathways, I want to just show this slide. These are the plant or naturally derived compounds that has been used for the different type of cancer cells that will exhibit the anti-cancer activity or cell death pathway to suppression of the FOXO3 activity or mediating through FOXO3 activity. If you take the drug such as called genistein that inhibits the proliferation of cancer cells by attenuating a negative effect of epidermal growth factor on tumor suppressor FOXO3, this genistein is nothing but a plant derived isoflavone mainly found in the soy product, tofu, and textured vegetable proteins that has been rich, that has been shown as a potential chemotherapeutic aspects through suppression of FOXO3 activity. The other plant-derived compound said to be 18-beta-glycerinic acid. It is called a pentacyclic triterpenoid found in Glyceria glabra roots, that is plant-derived component, that also induces apoptosis through modulation of this ECO-T FOXO3 beam in human breast cancer cells. The other study shows that an aqueous extract of Fagonica critica induces the DNA damage through cell cycle arrest and apoptosis via FOXO3. This Fagonica critica is nothing but as a potential therapeutic value for anti-hyperglycemic effect, such as anti-diabetic effect, that has been effectively published in several reports. So these compounds also has been used as an chemotherapeutic options. On the other hand, the purified Vituxin compound 1, it is called Vituxin negunda seeds, that is called aromatic shrub, that is abundant in Asian countries, also showing the apoptosis through FOXO3 in liver cancer. So these are all the already published reports which has been shown the plant compounds of the different model. Now coming to the other drug which has been used in this study is called, botanical name is called Becorides anthelmintica, and the synonym is called Conzea anthelmintica, Vernonia anthelmintica. This is the iron wheat seed plant. From this plant the seed has been obtained. So there are various names in India. It is called Katu Jigari, Kallu Jiri, Kal Juk, iron wheat. So these are the different names in different territory they are been calling. But it is ultimately bitter in taste, even though it looks like a zira that we are using for our regular cooking purpose, but it is very bitter in taste. Apart from this, we go to the cancer activity. These are the studies that has been shown several years before. What are the various activities apart from this cancer that has been targeted based upon this drug, Centranthum anthelminticum seeds? There have been shown anti-diabetic, antioxidant, diuretic, anti-helminthic, anti-filarial. Some of them are sold angiogenesis and wound healing, larvicidal effect, and antimicrobial agent, and melanogenics target, and anti-cancer and anti-inflammation that has been published. Now coming to the experimental design. So to elucidate the role of vernodolin, the vernodolin is nothing but an active fraction, which I am going to show in the next slide how we prepared or extracted or fractionated this vernodolin from these seeds. As an chemopreventive agent against breast cancer cells, the following experiments were carried out. We started from the in vitro MCF7 and MD-MB231 cells. First step, as I told, isolation and bioassay guided fractionation of vernodolin from the seeds. Cell cytotoxicity and RTC, a real-time cell growth analysis. These are the important factors for any anti-cancer drug. Before we designing, we have to fix this. And what are the cell death pathway that has been involved? Reactive oxygen species measurement and apoptotic studies, mitochondrial membrane potential studies, FOXO3 immunofluorescence and western blot analysis, and downstream analysis of PA3K AKT, and the siRNA silencing study which shows the FOXO3 nucleotides. If you take the in vivo model, we used LA7, that is called human breast cancer cells orthotropic model for the induction of a mammary breast cancer in the animal. And we used this vernodolin and the standard drug that has been used for breast cancer such as tamoxifen in both animal model. And the biochemical and tumor volume measurements, expression of tumor markers, PCNE, Ka61, phosphoFOXO3 and phospho-P27 in breast cancer tissues with Vanillin treatment. The objective of the present study is to study the Vanillin, the active fraction from the seeds, mediate the cell death, and measure the FOXO3 levels in breast cancer, both MCF7 and MDA-MB231 cells, to elucidate whether FOXO3 has any correlation in the downregulation of PI3K AKT pathway during Vanillin treatment, to study the Vanillin as any anti-proliferation activity in LA7 induced mammary tumor model in rats, that is in vivo study. Before going to that, I just want to show you what are the active instruments that has been used for this preparation and cell cytotoxicity analysis. The first one is called Hplc. We are all familiar with this. It is used to fraction high purity compounds. In case of large quantities also we can use, and required for subsequent evaluation, analysis and process in the shortest possible time by using different fractionation. The second one is called LCMS ITTOF. This is called a new type of mass spectrometer that combines the QIT and TOF analysis that used to fractionate and functionalize the active component that has been present in our fraction. For this biological assays, we used a cellomics eye content screening. It is called eye content screening analysis by using fluorescent microscopy. This is the automated method that is used to identify a phenotype or any substance that alters the phenotype of a cell in a decided method by integrated mapping, image processing and visualization of tools to extract data from each cell and cell population to determine a response. And fourth one we used as a real-time cell analysis, that is called RTCA, where a newly developed unknown drug where we can use here electronic cell array for dynamic monitoring of cell attachment, proliferation, damage and death. We can measure up to 100 hours. So these are the instruments we used specifically for the fractionation and the initial cytotoxicity analysis. Now coming to the methodology section, how we actively isolate the compound from the Centranthum anthelminticum seeds. If you see from this figure, where we can see we have taken 100 gram of seed of the Centranthum anthelminticum as I shown in the picture. It looks like a zera. This 100 gram has been nicely powdered and different solvent extraction initially has carried out, started with hexane, chloroform and methane, and all these extraction are initially done with the MTT analysis, and hexane and methanol extract doesn't show any cytotoxic activity of these fractions. But fortunately the chloroform extract at the 7 gram are showing the MTT assay, but showing that IC50 concentration on the breast cancer cells has been seen from these results. That has been further passed to the flash column, that is called the C18 column, where we able to get from the A, B, C, D, E, F 6 important fractions, and out of this you can see from these values we are mentioned here the fraction A and B. The IC50 has been shown between 5.5 to 5.8 microgram per ml itself, whereas the fractions C and D, E and F, even though it shows activity, the IC50 ranges from the above 50 such as 100 here and here above 40. So what we did is we recrystallized between the C, D, E, F, and importantly the Hplc column we used to prepare the fraction A and B further, and we can able to get from 10 mg purified colorless oil like compound from the 100 gram of seed. It is a colorless oil if it looks like a liquid, whereas the compound 2 we can also purify, we get a 183.5 that has been shown here, whereas the pure compound of the vanillin in the Hplc we can get a single chromatogram of the compound that has been prepared from the fraction A and B. So what we did further is we used, as I told, mass spectra analysis to identify what is the structure belongs to the vanillin. As I told in the previous slide, I showed two compounds. This is the pure compound which I shown 10 mg, where we can see the single peak, whereas the other compound which I have shown as showing the multiple peaks and also contribute very small amount of the vanillin. That doesn't matter. We further study, we focused on this fraction of A that has been having the single peak of vanillin. This is the molecular structure of this vanillin that has been confirmed through mass spec analysis. This mainly comprises of lactone and isoprene units, and this is called as a functional compound of sesquiterpenoids. This is the active component that has been present in the vanillin. So now we move to our R&D research activities, the data where the in vitro assay we carried out at two different cell lines such as called MCF7 and MDAMB231 cells. We all know that both are the breast cancer cells, but MCF7 has a P53 wild type, whereas MDAMB231 has a P53 mutant type. Other characteristics if you take MCF7 cell is it has possess a ER positive, PR positive and HER2 negative cells, whereas MDAMB231 cells as having the ER negative, PR negative and HER2 also negative. And this is our cell culture. This is a basic picture which give idea how we are going to do this. Now I will start with the vanillin, how it inhibits the proliferation of MCF7 and MBA231 cells by using both. Picture A is said to be a basic MTT assay, and picture B and picture C is said to be a real-time cell growth analysis. We all know MTT assay is a calorimetric assay for measuring cell metabolic activity. It is based on the NADPH dependent cellular oxidoreductase enzymes to reduce the tetrazolium di-MTT to insoluble formosome crystals, which has been purple color. So what happens? More viable cell metabolize MTT and producing the purple color. In the case of dead cells, it will become colorless. So we taken 3 different cell lines. MCF7 is a breast cancer model, MD-MB231 also breast cancer model, and normal primary mammary epithelial cell to show the toxicity effect of the drug. So what we observed, IC50 values between MCF and MDA is not major, becomes 6.2 to 6.5 microgram per ml of this against breast cancer cells. The good thing we observed is in the primary mammalian cells up to 18.7 it doesn't show the toxicity. So the IC50 of these on normal mammary epithelial cells is 18.7, whereas in the case of breast cancer cells it has been shown within the 7 microgram per ml. In further analysis, we are also taken the one upper concentration and other higher concentration for real-time cell growth analysis, where we can see the blue, pink and white and green up to 17. After 17 hours it starts slowly increasing the cell time analysis, and after 12.5, after 24 hours reaches, it started declining. But in the case of blue line, it goes up to peak of real-time cell growth analysis up to 24 hours. After that only it started decrease. So we observed up to until 100 hours. This is the best advantage of by using real-time cell growth analysis with the different morphologies, what are the conditions has been happening. And similarly, we are also carried out within MD-MB231 cells also. We can able to see up to 24 hours it is been stable. After that it started slowly declining. Whereas in the case of 12.5 and 25 in both the cells, it started declining immediately after it reaching the 24 hours. So based on these, we further moved with these how the nuclear cells or morphology of the breast cancer cells has been changed by using the different dyes. So this picture A will show the annexin 5 binding staining. We all know annexin 5 is a specifically binding to a phosphatidyl serine residue. This is a calcium dependent process. It is also called as an early event of apoptosis. Is the flipping of the phosphatidyl serine residue from the plasma membrane from the inside surface to the outside of the cells. So what happens in the control cells, we can able to see there is no fluorescence. That means there is no calcium that has been leaked out of the cell. So there is no fluorescence or less green fluorescence. But in the case of treatment with the vanillin, we can see the green fluorescence that has been excluded out on the cytoplasm of the cells due to the presence of annexin 5. When we measured the cells along with the nuclear dye that is called O-chest, it is fluorescence in blue in color that specifically binds to the DNA. So it is clearly evidence that annexin 5 that I can able to detect the apoptotic cell dead cells during the vanillin treatment. This is the quantification of this image. The other study is called as a Paladin staining. Paladin is a highly sensitive bicyclic peptide that is used to staining the actin filament that is called known as the F-actin. It is important role for cell stability and morphogenesis. So in the breast cancer study, we importantly analyze how the morphogenesis has been changing, and also it involved in many crucial process division, endocytosis and cell migration. If you take the Paladin in the control cells, there is no drug treatment so that the cell morphology remains stable, so that we can clearly see that nice morphology of the F-actin filament that has been present in the control cells. Whereas in the breast cancer treatment, this F-actin filament has been damaged. As I told in introduction, how the vincablastin has been blocking the mitosis and F-actin filament. Similarly, here also, the vanillin treatment, it ultimately results in the F-actin filament damage that will result in the less fluorescence. And this is to show that normal picture of vanillin fraction and along with the commercial available drug doxorubicin, how the nuclear morphology has been damaged, which has been shown as compared with the control. We can see the distinct morphology of the cells with intact DNA, whereas in the treatment with the doxorubicin, the commercial available drug, and along with our vanillin, we can able to see the nuclear fragmentation here. Now coming to the important aspect of other cell death process is called as a fluorescent activated cell sorting analysis of MCF7 and MD-MB231 cells. This is called four quadrants, we can say. It has been counterstained both with annexin 5 and PA. The lower left said to be in a control cell viable. Here is early apoptotic cells and late apoptotic, and here is the necrotic cell. So when we increasing the concentration of the vanillin starting from based upon the IC50, one is lower, one is higher, 3.125, 6.25 and 12.5, we can able to see nearly 32, and here the 22 and 44 and 32% of the early apoptotic cells, and the late apoptotic cells has been increased during that, increasing the concentration of the time. That has been observed both in MCF7 and MDA cells. These apoptotic cell percentage of quantification has been shown in this below graph. Now coming to the other important aspect said to be a cell cycle arrest, how it happening during the Vanillin treatment. This picture will show clearly that Z0, Z1 stage it has been inhibited. The control cells without Vanillin treatment, the Z0, Z1 value is 64, whereas the Vanillin treatment with the 6.25 and 12.5 both in MCF7 and MDA, which increases from 64 to 72.5, where 55 to 61, and also when we increase the concentration, there is a slight increase has been happening in the Z0 Z1 pace, 71.6 to 64.7. Another important factors in cell death process is called as the reactive oxygen species generation. How this Vanillin mediated reactive oxygen species generation has been carried out by using a commercially available dye called as H2DCFDA. It is an acetoxymethyl derivative dye that has been present on the cell membrane. Due to the presence of cellular esterase activities has been present inside the cell, these non-fluorescent H2DCFDA has been converted into fluorescent DCF. So these fluorescence green color has been able to capture during the fluorescent microscope. Where here the picture will show the control that has been stained with both the DCFDA dye and as I told earlier the DNA OHS dye that has been fluorescence blue in color, where the control we could not see the more green fluorescence here, so that when we merge we could not see. Whereas if you see the Vanillin treatment, both MCF7 and MDA cells, we can see the more green fluorescence. That indicates the presence of or generation of ROS generation during the Vanillin treatment in the breast cancer cells. The merged pictures clearly showing that increased generation of ROS both in MCF and MDA B231 cells. And this slide will show the bar graph average intensity of dihydrofluorescein estate stained in the nucleus. Further, how the Vanillin has the ability to carry out the membrane permeabilization, mitochondrial membrane potential and cytochrome C release. So here we did by using the cellomics high content screening by using 4 different dyes, as we see here: DNA dye, membrane permeability dye, mitochondrial membrane potential dye and cytochrome C release, and this final lane will show the clear merged picture of this all the analysis. So we carried out both MCF7 and MDA MB231 cells, the Vanillin treatment with 6.25 microgram per ml, where the membrane permeability in the control cells has been less, whereas mitochondrial membrane potential has been high. This is a normal happening during that condition, whereas if you see the membrane permeability has been increased, that results in the leakage of the cells during the Vanillin treatment in the cancerous cells, and also once the mitochondrial membrane potential has been lost, there is no fluorescence as compared with the control. It has been clearly indicates the Vanillin as the anti-cancer activity, and also which results in the more cytochrome C release into the cells, which has been clearly evident from this picture. This is a similar condition we can able to see in the MDMB231 cells, and this merged pictures which clearly shows that activation of the cytochrome C and loss of membrane potential and the both conditions. So further, how the Vanillin induces apoptosis through intrinsic caspase pathway and reduces the pro-survival molecule has been carried out by using a specific assay, calorimetric assay. So we measured here both the caspase 3, 7 and 8 and 9. The control cell doesn't have any showing any activity, whereas in the Vanillin treatment we can able to see the more amount of these 3, 7 and the executor caspase 9. Further, Western blot analysis has been carried out to showing the cleaved caspase 3 molecule 7 and 9 and PARP. PARP is nothing but polyadenosine ribosyl molecule that has been play important role, ribose polymerase that has been play important role in this execution of the apoptosis, where we can able to see the more amount of the expression of the protein during Vanillin treatment as compared with the control without Vanillin treatment. That has been evident in this Western blot picture. Now we coming to that FOXO3 and Bing relation during the Vanillin treatment. So far we have discussed about how this Vanillin will exhibit the cell cytotoxicity, how it creates the ROS, how it creates the apoptosis related activity through intrinsic pathway, and what are the mitochondrial membrane. These are the basic cell death aspects and the related results we have been seen so far. Now we have to tune into the how this Vanillin modulate the cancer cell pathway by using FOXO3 signaling. So what we did is, as I told in introduction, the breast cancer due to the cancerous condition, there is a lack of FOXO3 protein. This is called MCF7 control cells without Vanillin, and here MDF MB231 cells without Vanillin treatment. It has been evident from our study during the increased concentration of Vanillin that has been resulting in the increased expression of FOXO3 protein with a different concentration. We can see from here. On the other hand, the FOXO3 level has been upregulated in breast cancer condition, has been also decreased during the treatment. And the important factor that has been related with this BIM, that is related with the pro-apoptotic molecule that will lead to the apoptotic pathway, also been upregulated in both the MDA, MCF7 as well as the MDMB231 cells. So this will create some more interesting aspects for our studies further carry out in what type of signaling it has been played during the downstream pathway. These are the quantification of these respective proteins through Western blot analysis. Now coming to that, how this Vanillin play important role in the modulation of cell cycle proteins. As I told in the introduction slide, how the nuclear transport and other cell cycle proteins that has been happening between the normal cell and cancer cells. Here it has been clearly shown during the cancer cells, the P21 level and P27 level has been down-regulated, whereas in the treatment with the Vanillin it has been upregulated, whereas other cyclins related proteins has been upregulated in cancerous condition, also been evident in MCF and MDAB31 cells, whereas during the treatment with Vanillin, these cyclin levels has been down-regulated. So these are the important factors that has been play how this cell cycle proliferation and regulation has been happening between the normal and cancerous cell condition. So it has been clearly evident P21 and P27 has been upregulated during the Vanillin treatment. This bar graph will show that relative intensity of these MCF and MDAB31 cells of the different proteins. Further, importantly, we checked that how this FOXO3 nuclear translocation has been occurred in the MCF7 cells, because this is the most important protein that has been happening between the nuclear transport, where it has to be exhibit its activity from the cytoplasm into the nucleus. So important aspect we have checked is in the control cells without Vanillin treatment there is no fluorescence, less fluorescence we can say as compared with the Vanillin treatment. We can see the increased amount of the green fluorescence will indicate the Vanillin treatment results in the recruitment of this FOXO3 protein from the cytoplasm into the nucleus of the DNA. So that has been clearly evident along with the counter staining with Watchers staining of the DNA. Whereas the control cells we does not see any more or any meager amount of the FOXO3 expression. Whereas in Vanillin treatment we can clearly see that increased amount of the green fluorescence. And importantly, this study has been correlating with our standard drug that has been doxorubicin also we used in our study that has been commercially used, that shows the increased amount of the FOXO3 in the nucleus. Similarly, same condition, this experiment has been carried out to show the nuclear localization of FOXO3 in the MDMB231 cells also, where we can able to see the increased amount of green fluorescence when you treating with Vanillin with a different concentration along with the doxorubicin has been evident here as compared with control. One of the important compound is rather than fluorescent microscopy, we have shown in the previous slide how it has been translocated. Here by using in a specific nuclear and cytosol fractions that has been clearly show our hypothesis how this FOXO3 cytoplasmic and nuclear protein expression play important role after Vanillin treatment. As we know, in control without Vanillin treatment in breast cancer in the nucleus the levels will be low. That has been evident in both MCF and MDAB231. Whereas in Vanillin treatment we can see the slight increased concentration when increasing the concentration of Vanillin, where we can see the upregulation of these protein in the nuclear fraction as compared with an cytosol. And one important component we have to show here is the GAPDH and laminin B1, how purified nuclear fraction can be able to distinguish based upon this loading control. This is called the loading control, where the nucleus is very free of cytoplasmic content, so it does not show anything on GAPDH. Whereas in the case of laminin B1, the cytoplasmic fraction doesn't have any nuclear contamination, so that it can show only the nuclear fraction loading control. So this clearly indicates our fractions has been clean, and the preparation has been clear during the Vanillin treatment that results in the nuclear accumulation of the FOXO3 protein. Now coming to the important aspects, we all know that is the FOXO3 play important role and mediating the chemotherapeutic aspects in this Vanillin treatment. But what could be the happening if it is happening only through FOXO3 or any other factors also might be involved? To cross check or to confirm that, we used a specific FOXO3 silencing peptides during Vanillin treatment in MCF7 and MD823 cells, where the minus means control siRNA minus and plus, whereas this is specific FOXO3 siRNA minus and plus, where we can able to see in the absence of FOXO3 during the Vanillin treatment there is no such increase during the presence of Vanillin. That has been clearly evident as compared with the control, that is the standard siRNA. We can able to see the increased amount of FOXO3 protein. In the case of both MCF7 and MDA, when we silence a FOXO3 protein, there is no Vanillin activity through FOXO3 signaling that has been happening, has been evident from the silencing experiment. It clearly shows the Vanillin have the ability to activate a FOXO3 related signaling aspects. This is the percentage of apoptosis that has been carried out both in control and FOXO3 siRNA in both MCF and MD-MB231 cells. As I told in introduction, what are the downstream signalings that has been reported along with the FOXO3s? Most of the people have been studied the PI3K and AKT signaling. In our study also, we found that phosphorylated AKT level and phosphorylated PI3K level has been upregulated in cancerous condition both in MCF and MDA, and eventually during the Vanillin treatment these protein levels got downregulated, which can match us with our previous results. And similarly, there is no change in the total AKT that has been evident from these both the experiments. This is the relative intensity and quantification or densitometry analysis of these proteins has been shown here. This is the important assay called as a AKT kinase assay during the Vanillin treatment, and important substrate here we used as a GSK3 alpha / beta. This GSK3 alpha beta is an important substrate that has been used to measure the indirect measurement of the AKT kinase assay. It is also called as an immunoprecipitation assay, which has been used as an indirect measurement of the AKT kinase activity, where we can able to see that GSK3 beta substrate level has been increased during that cancerous condition that has been evident from both MDA and MCF7. But when we increasing the concentration between 6.25 microgram to 12, we can see the substantial decrease or depletion level of the GSK3 beta, which indicate the involvement of the AKT signaling that has been happening in the Vanillin treatment. So far we have seen how this results in the in vitro condition and what are the different cell death pathway along with the FOXO3 signaling and the downstream signaling pathway with the silencing. Everything we have been seen in vitro condition. Now we move to the results on the in vivo part by using an orthotropic model called LA7 mammary gland cells. There are several cancer types that has been used this orthotropic model. In our model we use the breast cancer cells, that is human LA7 cells, and directly injecting on the mammary fat pad breast tissues here. Apart from that, there are several studies they have been using the cells as a model, including brain, lung and colorectal. The main advantage of by using these LA7 cells in the orthotropic model is the time consumption. So within the 1 month we can able to wind up this breast cancer model, whether our drug is effectively can be used further in the clinical aspects that can be concluded from these animal experiments. So within the 30 days or 5 weeks of duration, we can able to wind up this animal model. Rather than chemical model, we have to wait until 3 months to induce the breast cancer in animals, 3 to 6 months. So this is the advantage why we are using the rat LA7 model for orthotropic condition. So what we did is, here the table clearly show the 5 different treatment groups. NC means normal control without any cells, means LA7 cells. Tumor control means only LA7 cells, and tumor control treated with a different dose of Vanillin. Started with the low dose 1 mg and 10 mg, and tumor treated with 1 mg per kg of tamoxifen. So here what we did is, we treated LA7 cells with the mammary gland. We are waited for until 2 weeks to develop a tumor. Once the tumor has been developed, intraperitoneally weekly thrice we given the dosage of Vanillin 1 mg or 10 mg to the animals for another 3 weeks. So the overall experimental week has been 5 weeks. So if you see the tumor control, due to the cancerous condition, the body weight has been decreased 213 as compared with the normal control without cancer cells. Whereas the tumor treated group along with Vanillin is slowly starting graining up to 10 mg per kg Vanillin, it reaches 221 as compared with the 213. And also if you take the tumor volume has been increased in the case of tumor control, that is called LA7 induced group 2143 size, whereas in the case of tumor treatment we can able to see the decrease in the tumor volume, and substantially 50% decrease has been happening in the 10 mg per kg body weight of the Vanillin, that has been matching with the tamoxifen that has been commercially used, such as 1 mg per kg body weight, that has been matching. And also the tumor reduction percentage we can able to see at the 1 mg per kg body weight, we can able to see the 44% of decrease, whereas in the tumor 10 mg we can able to see the 75% decrease, that has been matching with the tamoxifen, which has been showing 73%. Which has been further this study carried out on the proliferation markers, especially by using immunohistochemical analysis in the presence of diaminobenzidine as a substrate. We using a specific antibody called Ka67, it's a tumor marker, and proliferating cell nuclear antigen in a breast tumor tissue. What happened? In tumor control, we can able to see the more amount of proliferating cell nuclear antigen. This is the tumor induced animal tissue, and also the Ka67 level also has been increased, that has been shown the brown color spots along with the tumor control group. Whereas the Vanillin treatment with 1 mg per kg and 10 mg per kg body weight, we can able to see that decreased proliferation or expression of these Ka67 and PCNA markers along with, we compared with the tamoxifen. So overall our study showed during the cancer induced group, the tamoxifen or our Vanillin treated group are showing the less marker such as Ka67 and PCNA, which suggests this drug has a chemotherapeutic property. Further important aspect which I have been shown in vitro study, P27 keep1 and FOXO3 level also been downregulated. On the other hand, the FOXO3 level has been upregulated in cancerous condition, and P27 has been downregulated. That has been evident from our in vitro study. Whereas the treatment with the Vanillin, we can able to see the increased amount of P27 keep1 both 1 mg and 10 mg in the form of brown color spots, that has been indicating the presence of P27 keep1 in the animal tissue. This has been compared with the tamoxifen treated group also, we can able to see the increased expression of P27 keep1. On the other hand, the FOXO3 level has been upregulated more in the tumor group, that has been showing decreased or less amount of the expression during the Vanillin treatment, which has been substantially proves the involvement of the cell cycle regulator proteins. In final, I will show the schematic representation of Vanillin mechanism on FOXO3, PI3, AKT signaling pathway, how it happens in breast cancer cells. So what happens, this Vanillin will inhibit the phosphorylation of PI3K, and phosphorylation of this red, it indicates the inhibition. That will result in the inhibition of GSK3 beta, that will result in the recruitment of the FOXO3 protein inside the nucleus of the DNA, that will bind, that will upregulate the FOXO3 protein, that will ultimately activate the BIM protein, activate the BAX that responsible for the cell death. The ultimate end is the cell death process of the cancer cells. On the other hand, in the DNA, these upregulation of this FOXO3 protein simultaneously simulate the P27 CYP1 and P27 KEAP1 will inhibit the cell cycle regulated proteins such as cyclin E and cyclin D1, thereby it inhibit the upregulated or abnormal activity of the cell cycle inhibition or in the cell, thereby it mediate the chemotherapeutic aspect of the Vanillin during the breast cancer cells. In conclusion, this Vanillin inhibits the cell growth of MCF7 and MDMB231 cells through induction of cell cycle arrest and apoptosis. As I shown in the in vitro studies, it has been clearly showed apoptosis through ROS mediation and caspase activity. Further, these processes subsequently lead to the attenuation or stopping of MMP and cytochrome C release. Release of cytochrome C activates the caspase cascade and PARP cleavage to execute apoptotic program. and further the downstream pathways of PA3, AKT and FOXO3 pathways enhance the cytotoxicity of vanillin against breast cancer cells. These vanillin interfere with cellular process by targeting different intracellular molecules and apoptosis through a FOXO transcription factors and downstream targets such as BEAM, cell cycle checkpoint proteins P27, P21, cyclin D1 and cyclin E. So further the in vivo experiment with LA7 cells reveals the antiproliferative action of vanillin and confirmed by using a cell proliferative marker such as Ka67 and PCNA in mammary gland tumor model. So these vanillin used as a target for the therapeutic or preventive intervention in breast cancers as well as other cancer typologies. Most of my colleagues which has been collaborators currently working on this same type of drug with other cancer model also, which include the colon cancer, which has been effectively showing this anti-cancer activities. With this I would like to conclude my presentation. I am welcome all for your active participation, and if you have any clarifications, please feel free to write my personal email ID mentioned here as well as my official email ID, and thank you for your patience for listening this presentation. Thank you.